Okay, you are welcome. So in today's video tutorial, we are going to look at the polymerase chain reaction. Introduction to PCR. P stands for polymerase and C stands for chain and R reaction. So PCR is an abbreviation of polymerase chain reaction. So what is PCR? The PCR is a molecular biology technique that used to amplify a specific DNA segment. It can be a gene, it can be a marker, it can be even an exons or introns. So when you actually amplify or a molecular biology techniques that used to multiply or amplify a specific DNA sequences, that is what we call PCR. And of course, we know that in humans, we have about 25,000 genes. So you can decide to actually identify a particular gene and amplify it million times or copy it 100 times or 1 billion times. So how do you amplify? You amplify it using polymerase chain reaction, that is PCR. So that is the major aim of a PCR, is to amplify, is to make a millions copy or thousands copies of a specific DNA sequences. And it actually allows researchers to create millions of copies of the DNA segment from a small initial sample. With a small amount of the DNA, you can actually make, or you can create a millions copies of a DNA segment. Assuming now you are targeting insulin gene as a segment of DNA, or you are targeting FOX03 gene, which is one of the longevity gene. So with small amount of your DNA, you can get a billions copy or you can create a billions copies of insulin gene. You can get a billions copy of the FOX03 gene. So that is actually the major aim of the PCR, is to create a millions copy of a specific segment of DNA from small sample. So when we say small sample, means small amount of DNA. So this PCR is very important or is widely used in various fields, including genetics, forensics, and medical diagnosis. With the PCR, you can diagnose disease like cancer, like infectious disease, like malaria, typhoid. You can use PCR to diagnose patients with that. In fact, with PCR, a lot of diseases can be diagnosed with, and you can also use this PCR in forensics. Like, for example, if you want to know whether a particular species of living organism survive in one environment, you can get the samples from the soil sample. You can isolate the DNA and carry out PCR, and from there, based on the copies of that segment of the DNA, it would give you an idea whether that particular species of living organism lived on that environment or not. So PCR has so many applications, and very importantly, this PCR has steps. There are 3 steps, and these steps include denaturation. As when you have your DNA, you know that your DNA is double-stranded. So the first step is denaturation. That is to divide the double-stranded DNA into single-stranded. So you see, in this first stage of denaturation, you see that the DNA is actually separated into 2 strands. The 5' end, the 5' to 3' end, and then the 3' to 5' end. And this denaturation is achieved by heating the DNA to about a temperature of 94 degrees centigrade. So when you heat the DNA to a temperature of 94 degrees centigrade, the DNA template usually separated into a single stranded. It means that the major aim of the denaturation process at this temperature is to break down the hydrogen bonding that actually complement the two double-stranded DNA. Remember, hydrogen bonding is the bond that pair the nucleotide sequence in the DNA. So when we hit the temperature to about 94, so when we hit the DNA, the temperature of about 94 degrees centigrade, it will separate it into two, meaning that the temperature will break down all the hydrogen bonding that pair the nucleotides in the DNA. And then the next step is annealing temperature. So actually, when the temperature reach about 94 degrees centigrade and the DNA strand is separated, then the DNA will then cool down. The temperature will be reduced to between 50 to 60 degrees centigrade. And that is when the primers, look at it, the DNA primers will now anneal or will now bind themselves to each of the strands. And these primers are two. We have reverse primer and we have forward primer. So the reverse primer will now bind to 5' to 3' end, while the forward primer will bind to 3' to 5' end. So it is actually at this temperature when the temperature cools down, when the temperature decreases from 94 to about 50 to 60 degrees centigrade, then the primers will now bind. Then after the primers are now bind, then the next thing is an enzyme called DNA polymerase. So the DNA polymerase will now start adding the nucleotides. You see here after the primers are added, then the DNA polymerase enzymes will now start adding. It will now start synthesizing. The strand of the DNA by adding nucleotides. So it will be continue loading and adding nucleotides until when you have 2 copies of the DNA. So it means that these 3 steps of the polymerase chain reaction covered 1 cycle. So when denaturation, annealing, and extension take place, then it means that you have 1 cycle of PCR. And at the end, you expect to have 2 copies of your segment of the DNA. So actually, this is briefly on the steps of polymerase chain reaction, and we are going to look at one after the other in more details. So then the history of PCR. So the PCR was developed in 1983 by Kary Mullis. So Kary Mullis is the scientist that was able to develop this PCR, and he won the Nobel Prize in chemistry for this invention. In fact, this discovery of PCR method has revolutionized the area of molecular biology. It solved a lot of problems. So the techniques revolutionized molecular biology. It revolutionized molecular biology and genetics by enabling rapid DNA analysis. So the PCR method, you can rapidly analyze your DNA molecule. So it solved a lot of problems. It solved a lot of time, and it actually involved in so many applications. So early application of this PCR. So with the PCR, because it's from the PCR product, that is the product of the PCR, you can actually go ahead or you can proceed for cloning. So you can clone the segment of the DNA that you amplify. So that is why it is important in cloning, and it is also important in gene sequencing. So if you don't have idea of the exact nucleotides that form that specific segment of the DNA that are amplified or that amplified by the PCR, then you can further ahead to get the PCR product and send the sequencer. Then after that, you can also detect mutation. So after you do the PCR, then you sequence, then you can detect mutation. That is, you can check whether there is any change in the nucleotides in the gene. Like, for example, it is actually through this technique that scientists was able to reveal the hidden knowledge or the mystery knowledge of sickle cell anemia. So the gene is a hemoglobin gene. So you amplify the gene are amplified using PCR, and then the gene are sent for sequencing. So after sequencing, when the nucleotides are actually observed, they were analyzed. And that is when the scientist was able to detect that it's because of one single mutation the hemoglobin gene, and that is why there is sickle cell disease. So it is very important. The PCR is important in cloning, gene sequencing, and mutation detection. So this is actually the scientist that is Kary Mullis, the scientist that invented or that developed the methods. So then, what are the basic components of PCR? What and what do you need in PCR? So for you to carry out PCR, you need to have the following component. So you need to have your, let's say, your tubes. We have what you call PCR tubes. So for you to carry out PCR, you need the PCR tubes. So usually the PCR tubes is like this, and it comprises of many components. And this component includes DNA. So if you want to do PCR, so the first thing you need the DNA template, you add it into the tubes, and then you also need, of course, an enzyme. So this is the DNA template. So you add your DNA, and then the next thing you now also add your DNA. You also add your DNA polymerase enzyme. So add your DNA polymerase enzyme, and then you also add your nucleotide. So your nucleotide, adenine, guanine, cytosine, and uracil will also all be added. Yeah, will also are going to all be added. So after you add them, then you also add your primers. So you also add primers. So primers are also added. So this is what we call a PCR component. So for you to carry out this PCR, you need DNA polymerase, you need nucleotides, you need DNA, and then you also need primers. In fact, you also need you also need RNase free water. You also need RNase. You need RNase free water. RNase free water. Apart from RNase free water, you also need magnesium chloride because the enzymes, DNA polymerase require magnesium as a cofactor. So you also add magnesium chloride. So these are actually the component of the PCR. So you see the primers, the primer that we are talking about is actually two. Usually they come in fair, reverse, and forward primer. So the importance of these primers, they are actually a short sequences that initiate the DNA synthesis process during amplification. So they are the short sequence of the nucleotides that actually bind to the separated strand of the DNA to initiate or to start the PCR or to start the DNA synthesis. And we will look at it. And of course, we need the DNA polymerase, which is actually an enzyme that synthesizes new DNA strand complementary to the template. So the separated DNA is the template, and then the primers will now bind to the template. And after the primers bind to the template, then the DNA polymerase will now take in each of the nucleotides, adding on the template. It will be taking the nucleotides and adding on the templates. Then we will have the. So you see, this is actually, we can see clearly from here, this is also the component, as you can see. You see, you have the DNA sample, you have the primers, and then you have the nucleotides. And that, of course, you see, this is the PCR tube. You also have mixed buffer. So in this mixed buffer, that is where you are going to have all the components in this mixed buffer. You are going to have them. So like, for example, here, you have your DNA templates, and then you have the primers, which is reverse and the forward, and then you have the nucleotides. So these are the nucleotide adenine, thymine, guanine, and cytosine. So these are the nucleotides. And then these are the tag polymerase. So the DNA polymerase that usually used in this PCR method is tag polymerase. And then you have mixed buffer. So this mixed buffer will contain a lot of components. It may actually include magnesium chlorides and so many others. So you see, this DNA sampling, primers, nucleotides, tag polymerase, and mix buffer will all be added. It will all be added into this PCR tube. So after adding this in the PCR tubes, then this is the thermocycler. Thermocycler is the PCR machine. So we'll take the PCR tube and place it inside the thermocycler or the thermal cycler. So the thermal cycler is a PCR machine. That is the machine that you need to take your PCR tubes and add it. And that is where all the PCR process will start. So after you add the PCR mixture, this is the PCR mixture, then you now transfer it to the PCR machine. So after you transfer it to the PCR machine, then you are going to set all your conditions. So you see the conditions for this PCR, you see number one, the naturation is 95, and then you have annealing 55, and then you have the synthesis, that is extension at 72 degrees centigrade. So what happened is that, so after you take this PCR mixture or this PCR to contain all the components, then you add it into thermocycler, then it will now start. So the first cycle, the PCR process for the first cycle, at 95 degrees centigrade, the DNA will be denatured. You can see here that this is denaturation. And clearly see here that's okay, there is denaturation. So let's look at the denaturation. So you see at 95 degrees centigrade, you will realize that all the double bonds, all the hydrogen bonding, you can actually see clearly that all the hydrogen bonding, they have actually break. So that is why you see here, you have two strands. So after heating, you have two strands of the DNA, so it's separated. So after that, then the next thing at 55 degrees centigrade, that is after 95 degrees centigrade, then the thermocycle machine or the thermal cycler will now lower the temperature to 55 degrees centigrade. And that is where the primers will bind. So you see one of the primers bind here, then another primers also bind here. So you see, this is the 5' direction, and this is the 3' direction. And as you can see, that the primers have actually annealed, have bind themselves to the template. So they bind themselves to the DNA template. So after that, then the next thing is the synthesis of new strand. So the primers has already bind. So the next thing is the tag polymerase. It will now start adding the nucleotide. It will be adding the nucleotide, adding the nucleotide, and also here the nucleotide will be added, then until when you have a complete strand. So you see at the end you have two DNA, you have two molecules of the DNA. So this is what you call, briefly, this is what you call PCR method. So the naturation at 95, the double stranded of the DNA will be separated. Then the temperature will lower to 55 degrees centigrade. Then the primers will now annul themselves. And then the temperature will now go off, and that is when the DNA polymerase will be activated. Then the nucleotide will be added. Then at the end, you are going to have the double DNA. So that is on one side. So I can decide to ask you maybe, okay, so now in 25 cycle, how many DNA molecules do you expect in 25 cycle? So it means that in every one cycle, one DNA, in one cycle, DNA is split into two. So you have two DNA. Then in two, this is the number of cycle. So then in two cycles, then you have four, when you have four cycles. Then when you have 3 cycles, so each cycle produces 2. So if you have actually 3 cycles, then you have one cycle to another cycle to you have 6. So this is how it will keep dividing. So 1 divide into 2, and 2 divide into 4, and then 4 divides into 8. So it will continue like that. So you see, I think for this calculation, something is wrong. Yeah. Because let me now say this. You see, what usually happened is that in one cycle, you have 2 DNA. Then in the second cycle, that is 2 cycles, these 2 DNA molecules will also divide themselves to produce 4. And then in the third cycle, these 4 will divide themselves to produce X. And then in the fourth cycle, then these 8 will now multiply themselves into 8 will divide also into, because each one will divide into 2. So it will divide into 16. So it means that if you want to know the number of DNA molecules you are going to have at the end, I think if you can use 2 power n, you can get the answer. And then this n reflects the number of cycles. So if you have, let's say, 300 or let's say if you have 30 cycles, then how many DNA molecules do you expect? It means that you expect 2 power 30. So whatever you have here, it will give you an idea of the number of the DNA, or the number of the amplification, or the number of the amplicons. So this is how you can calculate it. So you can calculate it in this way. That, okay, the 2 power, the number of cycles, and that will give you the number of the amplicons. That is the number of the amplified DNA in the tube. So then let's actually look at the process. So PCR involves a series of temperature changes commonly known as thermal cycling. So that changing in the temperature from 95, 55 to 72 is called thermal cycling. And the 3 main steps in each cycle are denaturation, annealing, and extension. And each cycle typically doubles the amount of the target DNA, leading to exponential amplification. So that is why I said 2 power n. So it is exponential amplification. So then, so I think we explained this is the same thing as the previous. So you have your DNA template, you have your fragments, your nucleotide, you have your tag polymerase, and then you have your PCR buffer. Then you all add them here. And after you add everything here, this is called PCR2. Then you now take it to thermocycler, that is the PCR machine. And then you set your temperature. The naturation will start after you heat it at 94 to 95. It will denature and the DNA will be separated. And then the annealing, the primers will now anneal themselves, as you can see here. And then the tag polymerase will now start adding the nucleotides. And then at the end, you have the cycling, which will give you the number of the amplicons that you want. So then this is actually, let's look at the steps one after the other. So denaturation is the first step where the double-stranded DNA is heated to separate into 2 single strands. So you see, in this case, the DNA is heated to separate the DNA into 2 strands. And these steps usually occur around a temperature of 94 to 98 for about 20 to 30 seconds. So when you heat the DNA to around this temperature for about 20 to 30 seconds, the DNA will be separated. So usually we look at it here. So as you heat, this is actually, we call this melting temperature, TM. This is the melting temperature. So when you have your DNA and then you heat it, so as you heat it, the double-stranded DNA will be separated. So you can see this is the temperature. This is the temperature. The temperature at 90 to above, that is where the DNA separates itself. So this is the stranded DNA. This is actually the double-stranded DNA where it separates itself at a temperature above 90 degrees centigrade. So then, of course, the separation allows the primers to assess the single-stranded DNA template for the next step. So after the double-stranded DNA is separated, so that will allow primer to have access to the single-stranded DNA. So then for the next step. So the next step is the annealing. So therefore, during the annealing, the temperature is lowered to allow the primers to bind to the complementary sequence on the DNA. So when it binds, then the steps usually occur between a temperature of 50 to 55 degrees centigrade, and it lasts for about 20 to 40 seconds. And of course, the specific temperature depends on the melting temperature of the primers, because the temperature of the primers vary. So that is why if you want to know, especially if you are doing PCR, so if you actually want to know the best thing for you to have your amplicon, in most cases, the primers are coming with the template. When you have the template, you open the template and you read all the components. You read everything in the template. So after you read everything on the templates, then from there you can see the melting temperature of the primer. Like for example, right now I have one of the template of the primers where the melting temperature of the primers is 55.88 degrees centigrade, and that is the melting temperature of the primer. So you see, it is from this melting temperature that will give you an idea of what your annealing temperature will be. So the best thing now is since the range of the annealing is between 50 to 65 degrees centigrade, so the best thing to do now is to look at what temperature this primer annealed to this double-stranded DNA. So it means that in your thermocycler, you need to actually have your annealing temperature in the thermocycler. You have to set it around 56 degrees centigrade, and that is where you will have perfect alignment of your primer with the stranded DNA molecule. So that is why the specific temperature depend on the melting temperature of the primers used. So you see, if I have the melting temperature as 55.88 degrees centigrade, so the best thing when setting my annealing temperature in my PCR machine, I have to make it around 56 degrees centigrade. So then after this, the next thing that we are going to look at, of course, okay, let me actually show a diagram where you can see. So you see, this is the demonstration. So at 95 degrees centigrade, the temperature, at this temperature, the double-stranded DNA separated, and then the primers, which is the forward primers, look at it, the forward primers bind at 3' to 3' direction, while the reverse primer bind at 5' to, at 5' to 3' direction. So after the binding of the primer, then the next thing is actually the polymerase. So this is the top polymerase enzyme. So it now started adding the nucleotides. So until you have the final, until you fully synthesize the segment of the DNA. And this extension is usually at 72 degrees centigrade. So then extension. So in the extension state, the temperature is raised to optimal range for a DNA polymerase activity. So generally, enzymes work at a specific temperature. So the Taq polymerase enzymes work maximally at a temperature of 72 degrees centigrade. So if the temperature is below this 72 degrees centigrade, the enzymes will not work. And if it is above, the enzymes will also not work. So that is why after annealing, the temperature raised to 72 degrees centigrade. So then the DNA polymerase synthesized the new DNA strand by adding the nucleotides complementary to the complex. So the major work of the DNA polymerase is to add nucleotides complementary to the template. So these steps usually last for 30 seconds. It usually lasts for 30 seconds to a few minutes, depending on the length of the target temperature. So let's look at actually the synthesis. So you see, this is the reverse primer and this is the forward primers, and these are the nucleotides. So you see the enzyme is now adding them. It's now adding them in this direction for this template and also adding them in this direction for this template. So this is actually the 3 key steps in polymerase chain reaction, that is PCR. Then the next thing is the types of PCR. So there are different types of PCR. We have different types of them. We have separate types of PCR. We have several types of PCR. We have quantitative PCR, reverse transcription PCR, and then we have multiplex PCR. So the major work of the qPCR, it allows for the quantification of DNA. So if you want to quantify the amount of the DNA that you have, then you use qPCR. That one is for quantification. And this provides insight into gene expression levels. That is how much of the genes are expressed. Like for example, now if you want to quantify how much of the melanin are produced in your body, then you are actually going for the quantitative PCR, that is qPCR. So, and then we have multiplex PCR. So for the multiplex PCR is actually when you are trying to target multiple, when you are trying to amplify multiple target in a single reaction, saving time and resources. Maybe let's say you have in your DNA, you want to target 3 different genes. You want to target 3 different fragments. So you use multiplex PCR. Then you add all the primers, because in multiplex PCR, it means that if you are going to target 3 different segments of your DNA, it means that you are going to have primer for each segment. You are going to have primer for each segment. So it means that at the end, in your PCR tubes, you are going to add all those primers. So this actually is very important in saving time and also wasting reagent. So you see, these are the different types of the PCR. We have hot start PCR, we have real-time PCR, we have quantitative PCR, and we have digital PCR. So all these are the different types of the PCR method. And of course, they follow the same protocols, the naturation, annealing, and extension. And these are some of the applications of the PCR. You can use them for diagnosis, like, for example, detecting infectious disease like disease caused by microorganisms in research. You can also use them in research, that is amplifying DNA for cloning. So you can amplify DNA for cloning, and you can also use it in forensics, that is analyzing DNA in criminal investigations and paternity testing. So if there is any criminal case, you can actually get the DNA and and use it to investigate the criminal cases, and also the paternity or maternity testing. And then also drug development. So screening for drug targets, drug efficacy, and pharmacogenomics. So you can actually use PCR. So PCR is important in identifying drug targets and also trying to actually know the drug efficacy, that is the efficacy of the drugs, as well as pharmacogenetics, and also environmental monitoring. So identifying microorganisms in soil, water, and air samples. You can also use PCR to do this. So these are the different types of PCR, and these are some of the application of the PCR method. Then application of the PCR, although I have actually mentioned them there. So PCR is used in medical diagnosis to detect pathogens, that is infectious disease, to detect mutations, to detect hereditary diseases like sickle cell anemia. It also plays a crucial role in forensic science for DNA fingerprinting and evidence analysis. And also PCR is also used in environmental biology to study biodiversity and microbial community. So all these applications is similar to these ones that I just mentioned. Yeah, so ladies and gentlemen, this is where we are going to stop, and I hope this video will be very important to all of you. And please don't forget to subscribe and join my YouTube community for more videos. Thank you very much. See you in my next